Hello everyone. This is Jaymin Patel, Assistant Professor, Department of Pharmaceutical Technology. Welcome to the MOOC series, an initiative taken by the LJ Institute of Pharmacy. In this session, we will discuss about immunization. So what is immunizations? So immunizations is defined as a procedure by which the body is prepared to fight against the specific disease. Okay, in the immunizations, main the two type of immunizations. One is the active immunizations and second one is the passive immunization. In the active immunizations, antigen is injected in the body, and in response to that antigen, the antibody produced by the host body. While in case of the passive immunizations, the host body immune system is compromised. So that's why there is need to supply the antibody itself to the host. Okay, so here the first one is the active immunizations. Okay, so preparations, okay, that provide the active immunizations is nothing but the vaccine. So what is the vaccine? So vaccine is the substance that is introduced into the body to prevent the disease produced by the certain pathogen. In the vaccines preparations, okay, maybe the dead body of microorganism is there, or maybe the live attenuated microorganisms. Now what is the meaning of live attenuated microorganism? Means this microorganism is artificially treated in such a way that is remain live but is not capable to produce the disease. Means that toxicity is completely destroyed but that antigenicity is maintained as such. Okay, so this type of preparations is used because in live sense, okay, sometimes our immune system respond quickly and responds better way. Okay, so that's why the live organism is utilized in some of the preparations. Suppose for example polio drops is there, okay, in that the live virus is present. So vaccine that produce the immunity against the pathogens, okay, by the two mechanism. One is by the activations of the B cell, and that B cell is going to produce the antibody, and second one is the activations of the T cell. Okay, it will produce the cellular immunity. Okay, so here if we talk about the type of the vaccines, if I talk about the type of vaccines, okay, so main four types is there. First one is the live attenuated vaccines. Okay, live attenuated vaccines means is contain the modified strain of the pathogens. Okay, it may be bacteria, it may be virus. Okay, is weakened by the special treatment. Okay, so they are able to multiply within the body but is not capable to produce the disease. Okay, is just capable to provide the antigenicity, and due to this antigenicity our immune system is activated and it will produce the antibody relative to this microorganism. Okay, so live attenuated vaccines. Second types is the kill inactivated bacteria. Okay, suppose for example after the death, okay, if the protein structure of the bacteria is remain as such, so generally we destroy the bacteria, that is we destroy the microorganism by the special treatment. Okay, it may be the heat treatment, it may be the chemical treatment, and this dead body of bacteria is utilized to prepare the vaccines. Okay, so vaccine is nothing but the suspensions of dead body of the bacteria, and it will provide the antigenicity to the our immune systems, and our immune system will produce the antibody relative to this bacteria. Okay, third type, that is the subunit vaccine. Okay, it is the most interesting vaccine nowadays, and the side effect due to these subunit vaccines is minimum. Okay, so how this subunit vaccine is prepared? So in the subunit vaccines the whole microorganism is not utilized, but some specific part of the microorganism that is act as the antigens. Okay, okay, we already discussed the terms epitopes. What is the epitopes? Epitopes means the site. Epitopes means a site that is present over the surface of the bacteria where the antibody is going to bind. So that epitopes of the microorganism is utilized to prepare this vaccine. And here means it is nothing but the special part of the bacteria, is the special part of the bacteria, the whole body is not utilized. So here whatever the side effect associated with the other part of the body, okay, that side effect can be minimized. So see here the subunit that contain only the essential antigens, okay, and not all other molecules, okay, that make up the microbes, and chance of the adverse reactions of the vaccines is comparatively decreased. Okay, if we talk about the type of subunit vaccines, so toxoids is there, protein vaccines is there, recombinant protein vaccines is there, polysaccharide based vaccine is there, and conjugated vaccines is there. Okay, nowadays, okay, whatever the vaccine, okay, maximum research is going to generate the vaccines for the COVID-19. Okay, so it's nothing but the one type of subunit vaccine. Okay, Zydus Cadila and Bharat Biotech of our India, okay, is work toward this vaccine preparation so is the DNA based vaccines. So it is not a means in this preparation we are not going to use the whole body of the virus. We use the specific gene sequence of the virus, and with the help of this gene sequence we are going to prepare the vaccine. So it is nothing but the one type of subunit vaccine. Now the fourth one, that is a combination. Okay, it is not a new types, but here in the combinations vaccine we are going to combine the two or more than two antigens. Okay, so by the single dosing, okay, that host, otherwise that client, otherwise that child receive the immunizations against the two or three antigenic agents. Okay, so here we are going to mix. Okay, if this all the antigenic agent is compatible with each other, then we can go for the combinations of this all the antigenic agent. Okay, so advantage of this combination vaccine, that is the simplified the administration. Okay, suppose if the three antigenic agent is mixed in this preparation, so ultimately, okay, if we combine, so by the single dose we provide the immunization to the patient. Otherwise if we give the patient to the patient means if we give the child, then it is required to give by the three times. So simplified administration is that by the single dose we provide the immunizations against the three antigenic agent, reduce the cost, okay, improve the timeline of the vaccinations, and reducing the storage uh, problems. Okay, so here example of the mixed vaccine, that is the DPT is there. Okay, diphtheria, pertussis and tetanus. Second one is the DT, diphtheria and tetanus is there. DP, diphtheria and pertussis is there. And MMR means measles, mumps and rubella. Okay, so these are the example of the mixed vaccine. Okay, here some classification is given. Okay, in the live attenuated vaccines, example is the MMR is there, varicella is there, influenza is there, rotavirus is there. If I talk about the killed, so in that polio is there. Okay, in the polio the two type is there, polio vaccines. Okay, two type of polio vaccine is available. One is the injectable polio vaccines and second one is the oral polio vaccines. Nowadays the maximum oral polio vaccine is utilized. Okay, so this injectable polio vaccine is the killed form, while the oral polio vaccines, okay, means the do bund polio ke. Okay, so this oral polio vaccines is a type of live vaccines. Okay, so here next one is the hepatitis A. Third one is the combined vaccines, that is the DPT, NTT and subunit conjugate means the hepatitis B is there, influenza is there. Okay, pertussis, diphtheria, tetanus, this is also available in the subunit form. Now the next topic is history of immunizations. So how this vaccine is invented? So first vaccine is prepared by the Edward Jenner. So on that time, the maximum person is infected by the smallpox disease. So this Edward Jenner tried to find the treatment solutions for this smallpox. So he observed that one of the persons, that is a milkmaid, okay, is remain protected against this smallpox. So when he take the interview, so on the basis of that he come to know the cow of that milkmaid is already suffered by the cowpox. So that person, that milkmaid is exposed to this cow many times, so the virus responsible for the cowpox that enter inside the body, this virus, in response to this virus the immune system is activated, it produces the antibody, and that antibody is capable to protect that person against the smallpox. So this virus for the cowpox is not capable to produce the disease in the human, but is capable to provide the immunizations to the human against the smallpox infection. So here the first preparation prepared by this Edward Jenner from the cow, and in Latin terms the cow is called as a vacca. So that's why that first preparation is called as a vaccine. So vaccine term is utilized for that preparation only on that time, but later on world accept this word, okay, for all the preparations that provide the preventive measures, that provide the protections against the infections. Now if we talk about the steps of the vaccine preparations, so here first step is we have to select the suitable strain for the vaccine production. Okay, so if you go for the live vaccines, otherwise if you go for the dead vaccines, so according to that we have to select the suitable strain, that strain. Okay, if you go for the dead vaccines, the same strain that we will use, and if you go for the live vaccines, then we can go for the alternative strains is capable to provide the immunization but is not capable to produce the disease. Okay, so first step is to select the strains. Second one is the growth of microorganism. Okay, we will subject this strain into the suitable culture medium. In this culture medium, the growth of this microorganism is like a sufficient microorganism is produced. Later on, we will go for the isolations and purifications of this microorganism. Okay, so that all the microorganism is isolated. Next stage is the inactivations of this organism. The inactivation is done by the two way, by killing, otherwise by reducing that toxicity. Okay, inactivation is done. Later on we subject for the formulations of the vaccines. Okay, in the vaccine preparations we add the different additives. Okay, we add the stabilizers, we add the suitable surfactant agents. Okay, we add the different additives into the vaccine preparation, and final stage is that is the quality control test, and finally that vaccine preparations is entered into the market. Now here, these vaccines should be stored in suitable temperature. So if we talk about the storage of the vaccine preparations, okay, so vaccines is nothing but the protein based product, so is highly sensitive to the heat. So that's why the cold conditions is required to provide to the vaccine preparation. So from the manufacturer to the client, the vaccine that move from manufacturer to the client, till that vaccine should be stored in suitable temperature. And this storage system from manufacturing to client is called as a cold chain system. So cold chain is the system of transporting and storing the vaccine at recommended temperature, at cold temperature, from point of manufacture to point of use. Okay, so in the cold chain storage equipment, main three equipments is used. One is the walk-in cold room, second one is the deep freezers, and third one is the ice-link refrigerator. Okay, here if we talk about the stability, so some of the vaccines is highly sensitive to the heat, and some of the vaccines is less sensitive to the heat. Suppose if we talk about the OPV, that is the oral polio vaccine, is the oral polio vaccine is highly sensitive to the temperature. So that's why it should be stored into the very cold temperature, very cool temperature. Okay, it should be nearer to zero. Okay, so OPV is there, measles is there, BCG is there, MMR, hepatitis B and DT. Okay, it is the descending order. Okay, means here hepatitis B and DT at the bottom is less sensitive to the uh, heat. So that's why if we store at a cold condition, means if the temperature is 4 or 5 degrees Celsius, it's okay, it is suitable for this vaccine preparation. Okay, if I store the hepatitis B is there, BCG at very cold temperature, means the freeze dried temperature. Okay, if I store at a minus degree temperature, then these vaccines will destroy. Okay, so these vaccines, okay, hepatitis B is there, influenza is the BCG, it should be stored at a cold temperature, means 5 to 10 degrees Celsius. While in case of the OPV is there, measles is there, it should be stored at a chilled temperature. So here the sensitivity of the different vaccines is different toward the heat. So according to that we have to store that vaccines. Okay, some of the vaccines is the light sensitive. Okay, for example OPV is there, measles is there, MMR is there, varicella. So this type of vaccine should be stored in such a packet where the light should not enter, and that packet should be opened directly in front of the client. Okay, so vaccines should remain protected, vaccine should remain protected against the light. Okay, here the storage, if we talk about the storage, in some of the small clinic the vaccine is prepared, vaccine is stored in normal, the common fridge. So, and in that fridge the food component is also present, so vaccines should be stored in the dedicated fridge. Okay, vaccine should not be stored in the normal fridge uh, in the clinic. Okay, so this is about the storage of the vaccine. Now, how we can check whether this vaccines is properly stored or not when it reach to the patient? Okay, how we can come to know this vaccines is properly stored or not? So, on the surface of the vaccine preparation, okay, means whatever the vials is there, on the vials one VVM monitor is there. Okay, okay, you can see here the picture VVM monitors. Okay, so here in the VVM monitor, okay, in the center the square is there, that square is light in color. When exposed to the heat, that color shade is going to decrease, that lightness is going to decrease, and internal square is become dark. Okay, you can see in the picture the internal square is become a dark when it exposed to the heat. to the maximum time. Okay, so here our vaccine topic is completed. Thank you very much. Thanks for watching this video. For more updates, like, share and subscribe our YouTube channel Pharma Ignites. Press bell icon for the notifications.